All right. Hey everyone, and welcome to our second guest lecture. So we're going to switch gears a little bit and talk about diffusion models for protein design. And for that, it's my pleasure to introduce Jason. Jason is a PhD student supervised by Tommy Akala and Regina Barcelay right here at MIT. And he really has been a leader in the development of flow and diffusion models for protein design and structural biology. His models developed here at MIT and then also co-developed with recent Nobel Prize winner David Baker have been widely used in structural biology. And you're going to hear that really what the potential and power of these models are. So thanks for coming, Jason. Thanks for preparing this talk. All the best, and we'll do around like 30 minutes of presentation, and then I think we're right on time, so it should be fine. I think you have to turn it on. Yes.
Okay. It's on now?
Yes, it's on. All right,
okay.
Cool. All right, let's give him a round of applause as a welcome.
Right. Okay, yeah, thanks Peter for the introduction. So I'm going to be talking about diffusion models for protein generation and then downstream for protein design. If you have any questions, feel free to just say them while I'm talking as well. Definitely have more slides than needed, but hopefully we'll get through the important parts. So I guess I'm going to assume that people don't know what proteins are, but you probably know a little bit about it from biology. But I'm going to go through like some of the basics and to motivate the problem. I think, as you know, kind of in biology, proteins kind of carry out a lot of the functions for life. One of the simplest things that they had to do is like DNA replication, where just making like 2 cells, you have to create 2 copies of a DNA strand. And proteins, they're actually the molecular mechanism of how this works. In DNA replication, here's like an animation of how proteins kind of work together to create new DNA strands. And this is kind of a very complex process that nature has taken millions of years to kind of come up with. But kind of unlike nature having like billions of years, as humans, we're creating a lot of issues for ourselves, like a lot of pandemics coming about, as well as creating too much plastic, and then even our own biology causing diseases that we have to rely on genome editing, like using CRISPR-type systems to solve for us. And so as humans, we've kind of engineered proteins, molecules to become drugs for specific needs for us, but this has to be done a lot quicker than the timescale of how nature comes up with new functions. So you might ask, like, okay, we've seen vaccines, like when SARS-CoV-2 happened, we came up with like vaccines, so we kind of like technology to do some of this stuff, but why do we need AI? And I really like this figure to kind of illustrate the fundamental issue and like kind of why AI is important in this space. I think, as many of you are probably computer scientists, Moore's law says that across time, the constant transistors should like go down exponentially. So then the cost of new compute like dramatically decreases. And that's kind of led to the computing revolution and NVIDIA's like stock market prices and stuff. But then in biology, we have the opposite. And so Evrum's law is Moore's law spelled backwards because the opposite happens. Over time, the cost of drugs doubles every couple years, and so there's like logarithmic scaling of drug costs. And so there's this crisis actually in the healthcare where it takes maybe like around 10 years and about $2.6 billion to even make like a single drug, which is kind of insane if you think about it. We have so many diseases in human health, like there's the bird flu now and stuff, so imagine like we don't have eggs that cost $2 again in 10 years. That's kind of crazy to think about. So kind of can AI actually accelerate this timeline so that yeah, we can find drugs a lot faster? So I'm going to talk a little bit about protein modeling to kind of make sure we have like this background knowledge. DNA, I think as we all know, is just like 4-letter vocabulary. And then from DNA through the central dogma, that becomes like a protein sequence, which becomes a structure. And then the structure is kind of what we can look into and try to figure out like what the function of the protein is. And each of these is like a different data type. You start from like a sequence that has a 4-letter vocabulary to 20. To the 3D coordinates. So it's also this very multimodal problem as well. You have to care about both this discrete type of data structure and this continuous type of data structure. And in function is this kind of like Wild West that you also have to care about dynamics of how proteins move and stuff. And the kind of the way that I like to think about it is, let's see if this pointer works. Yeah, DNA is kind of like the assembly language of biology, and then like proteins is like the Python of biology. And when you're trying to design stuff, it's in code, it's much easier to work in Python because it's like a much higher level thing that's closer to what you actually want to do. So that's why a lot of biologists like to work in protein space because it's often easier to work with. And kind of what is protein function, if you aren't familiar with it. This is kind of how a virus works. A virus will infect a human cell, and then it'll replicate itself, cause the cell to die, and as the cells die, we get sick and sometimes even die because of that. How do protein binding style viruses? You might try to design like a protein that's called an antibody, or your body might make them itself that'll kind of attack these viruses. So it'll kind of like surround them and tell the immune system to stop it from infecting a cell. So if we can have like an AI algorithm that can come up with these antibodies kind of almost immediately, then we kind of have like a one-button solution to kind of kill any virus that might appear in the near future. So this is a very simplified example. There's a lot of nuances here, but that's kind of the basic of kind of how we might want to use this technology to create new vaccines and drugs. And I think a lot of us have kind of known or kind of seen in the news a lot of revolutions in proteins and AI. The one that may be most familiar is AlphaFold 2, which was an algorithm developed by DeepMind, where you go from like the protein sequence to predicting the structure. And this kind of was in 2021, that kind of revolutionized AI and biology, and a lot of people have been pouring into this field since. But then one other algorithm that might have not gotten so much attention is actually going the inverse way of going from a structure to a sequence. And this is actually a generative model kind of based off a graph neural network that uses like an autoregressive generation along the structure. And then also very recently, something that I've been involved in was RF diffusion, which was given like a function such as binding to get the structure from that. And so you can kind of see that I call it very specifically because this kind of creates like a pipeline of how one might go. Damn it. How one might go from sequence to structure. And like these developments have also led to the Nobel Prize in Chemistry last year. So David Baker for protein design, and then Demis and John at DeepMind for the protein structure prediction problem. And kind of the time frame, like if you imagine that AlphaFold 2 was released in 2021, and this award was only given 3 years after AlphaFold 2, it kind of just shows the impact of these algorithms and what sort of impact they had in biology. It's just kind of, like also Geoffrey Hinton won a Nobel Prize in the same year, really shows the impact of these algorithms so far. So in the generative de novo protein design pipeline, these three algorithms have kind of led to this pipeline that's been really successful in protein design. You go from a function to a structure to a sequence, and then you use AlphaFold to kind of filter and figure out the final designs. And this is kind of like a little bit more of a illustrative schematic where I might want to tell the model, here, generate me a protein that binds right here. I'll create a structure that might do that, and then I'll create a sequence that'll hopefully become that structure, and then I'll use AlphaFold to kind of filter out which ones I want to end up using. And so I get asked this question a lot, which is that why do you need to know the sequence? But the sequence is the ultimate thing that we actually want to get down to so that we can actually produce and synthesize the protein. So this pipeline has been very successful, and kind of like before 2022, the structure generation was the rate limiting step, but diffusion models is kind of technology that has enabled this step in kind of what my research has been about. So I'll be talking about this for the next 40 minutes or so. Okay, so I think the last thing to say is like if you're like entrepreneurship-y, stuff like that, generative AI is having a huge impact in startup space. So recently there was a startup by David Baker's lab, actually based off RF diffusion, that is backed by $1 billion. There's also like isomorphic labs, the spin out by DeepMind, where they have like these crazy billion-dollar deals with pharma companies. And also there's like this language model-based technology for protein design that recently raised $142 million. So this is like also a very active space for entrepreneurship if you want to work in AI and technology. So yeah, last thing I'll say before I go into actual technical details is kind of like what is the overall goal and kind of what we actually want to get to. And this is kind of how I view it, is that we want generative AI to create these molecular libraries of proteins for us. And then we want to be able to use the experiment and optimize the experimental outcomes for them. So you can also think of this as like post-training or fine-tuning from like how OpenAI trains its models. I think if we can unlock this, where we can actually connect the biology to the AI and have this iterative loop process, it also unlock a lot of things in biomolecular design that will enable new drugs and stuff. So the overview of what I'm going to talk about, I'll first go over like protein structure generation using diffusion models, some of my own research. And then once we have this kind of basic algorithms of how to do structure generation, we've utilized them for downstream protein design tasks. And something I probably won't get to, but I'll just highlight, is we can also do this like multi-space, both discrete and continuous space generation of proteins, and then just go over some outlooks at the end. So kind of the desiderata that we want is that we want an algorithm that can generate, for one, high-quality structures, but also a distribution like diverse, also novel structures, and also things that actually function and work. Yeah, so kind of like how do you actually model a protein structure? It is basically just a collection of coordinates in 3D space. And so you might think, okay, the very naive thing is let's just take the diffusion models that like Peter has gone through and like with Gaussian noise and just apply them to every single coordinate here. Now, the issue when you do this is that because proteins have a lot of constraints to them, that they have to like satisfy this like biophysics. They can't be clashing with each other just because of physics. And there's like this bond geometry that they have to satisfy. That it's kind of hard to control the generation of these proteins because they're always like breaking and stuff. And so we actually had a paper in 2022 that tried this very naive idea of just applying Gaussian noise and trying to figure out like every single coordinate through diffusion. And it kind of worked. We also got into like a conference, but there were some issues with it that it was kind of difficult to scale and the performance wasn't so great for what we want to accomplish. That said, one thing I think has recently shown is like the bitter lesson is that if you actually scale them to like industry level, kind of like how DeepMind or Google has, then you can actually learn like non-equivariant architectures and methods that can generate proteins with pretty high accuracy. So that's like one option to do it. But another option is that you can try to respect the geometry of a protein, where there's like a linear chain of like how the atoms are connected to each other. You can respect this pattern that happens between every atom in a protein, and then you can kind of just do diffusion over the angles in the angle space. And so this kind of respects the geometry a little bit more, but it leads to this odd effect, which is called the lever arm effect, where if I change the angle just a little bit in one of these residues, then it's going to cause this like humongous change in the protein that's going to cause very unstable dynamics when you optimize or do diffusion over this. And so because of this, you are respecting the geometry by keeping the bonds fixed, but it's kind of harder to control the atom placements. And so one thing that we developed was to, like something that's like in the middle between both of these, is to model this frames along a chain. And I'll go into this a little bit more detail, but this is in the best of both worlds, where that you have most of the bonds fixed, while you also have like precise control over like where the frames go. And so this is kind of going into like biochemistry a little of like looking into how a protein is actually constructed. But if we just like zoom into the atoms here into this little space, I've already kind of mentioned that there's this pattern of these 4 atoms that are kind of repeated throughout the chain. It's just kind of like flipped here and is like also repeated there and stuff. We can realize that these 4 atoms are usually very rigid, and then we can represent them by an element of SC3, which is like a special Euclidean group that has both a translation component and then a rotation component. And that's just constructed by taking the 2 vectors coming out from the central C alpha coordinate to the other 2 coordinates and then doing Courant-Schmidt over that. So by doing this, you kind of have this like rigid representation that's keeping the bonds fixed, but also letting you move this around anywhere by just moving the C alphas in a translation. Yeah, so any questions about that before I move on? Or good? Yeah, sorry, go first. Yeah. So the scaling issues, we were trying these like graph neural networks. We weren't trying fancy like transformers. I think some of like the transformer scaling models kind of came out after or kind of around the time of this paper. So we didn't try any fancy scaling architectures. Also, we were kind of on MIT Compute, which isn't Google Compute as well, so couldn't try scaling it too far. But there have been like papers like AlphaFold 3 that show that you can scale it to Google Compute and achieve some of the scaling results that can just get rid of equivariance in there. AlphaFold 3 for the diffusion, they use a transformer there. They kind of use the diffusion transformer architecture that kind of like Sora and like that they use. Yeah, so kind of AlphaFold 3, they kind of like threw out equivariance completely, just be like, let's just do data augmentation and use a big transformer, and it kind of worked. I guess that's a good question of if transformers is the necessary ingredient, if that's sufficient and necessary. And I think that's like open research that everyone's looking into right now. Yeah. There's one more question. Yeah, that's a good question. Yes, proteins can scale to like tens of thousands of atoms, which causes a big issue. I think there's been a lot of work in like scaling transformers and stuff. So transformers actually scale pretty well with new tricks like flash tension and stuff like that. They scale like subquadratically, I think. But a lot of protein equivariant architectures do not scale that well. So this is one issue why grouping the atoms together, like how I did here, is one way to get around that, but you're only kind of saving a constant factor when you're grouping atoms together. So you have scaling issues either way, basically. So you could do like smart ways of doing like local attention or like grouping attention together to batch things up, but there's a lot of tricks to get around it. Okay, thanks. Yeah, and then so diffusion for protein frames, like where we kind of group the atoms together, kind of looks like this, where we start with noise on the rotation and the translation elements, and then we want to kind of go between noise and data in this space. So the forward process, it's governed by this stochastic differential equation, which I think you all are familiar with, and then you have this reverse process of sampling, and the thing you actually want to learn is this score term, which, yeah, by neural network. And so I think one thing to highlight here is we went about doing diffusion over Riemannian manifolds. You all are probably familiar with diffusion over Gaussian spaces in real space with the Gaussian distribution. But then here we have to figure out how we're going to do rotations over something that's not Gaussian, like SO3. So yeah, I just have this process of like how forward diffusion actually looks as you iteratively add noise to the protein. And then this like next slide kind of goes into detail, a little bit of detail about kind of a little bit of the math, where Brownian motion can just be described by this Gaussian kernel, but then in Brownian motion, you have to kind of project a Gaussian onto the manifold of SO3. So that causes this unique distribution called the isotropic Gaussian on SO3. The formula is like this infinite exponential sum that I'm not going to write here, but one thing to keep in note is that there is this translation between like in differential geometry of the logarithmic and the exponential map that you kind of have to take care of. But if you take care of all the math pretty quickly, if you take care of all the math quite rigorously, then you end up with something that has the same intuitive flavor as diffusion on Gaussian space, where at time 0, which is like your data, the distribution is going to be centered at your ground truth, and as you add more noise to it, it's going to start kind of sampling around the manifold until like at time t is going to be sampled uniformly around this manifold. So I think just in a second time, I'm not going over the mathematical detail here, but it's all in the paper later on. And so what we call frame diffusion is that we parameterize the proteins this way, and we're going to corrupt them with the noise that I described before, and you can train a model to uncorrupt this noise to get back to the original state. And then starting from pure noise, you can be able to use a neural network to sample the data iteratively here. And in the model architecture, we heavily rely on the AlphaFold2 architecture, where in proteins, you care not just about like attention over the tokens, but you also care spatially that protein, like each residue, each frame should kind of attend to things that are local to it. Kind of want to have this local bias, but also allow for these global interactions. And so the spatial attention is kind of complicated, so I'm kind of leaving that as like a black box. Positional attention is just like a transformer architecture where you kind of add this in here to, you add self-attention here to kind of help it be able to attend to neighbors that are in a long-range interaction. And a neural network will kind of iteratively update its embeddings using both positional and spatial attention to get the final embedding. And then the important thing is invariance as well. As you know, like proteins, if you rotate them, then they kind of have to have the same function. So you want to keep this SC3 invariance when you're sampling from this diffusion model. So there's a few things for this to satisfy this invariance criteria of your diffusion model. Is that one, you want to have an equivariant score model so that your score will like rotate if the protein rotates as well. And then you also want to keep the translation invariance by zero centering at each time. And so, yeah, this is kind of like in the algorithm here where you just want the score function to be equivariant. And so the goal is that like if I start from some noise, sampled noise, and if that ends up being decoded into this protein, I might rotate this noise, and also the final protein might be rotated as well. And this should be the same protein, have like the same likelihood under the distribution that your diffusion model eventually learns. And so this is kind of what the generation process looks like. That's starting from pure noise. No other condition is that just generate me like a random protein. And then the protein, and then the model is going to, iteratively decode it, and it's going to come up with like a diverse set of proteins here. And so kind of how to check if these proteins are good or not is something called, I'm going to call it like a realism check. There's some other names out there, but just for like layman terms, we're going to see if like a sequence could exist that could actually fold into this AI-generated structure. So let's say like this new protein is generated by the diffusion model. We're going to compute a sequence for it, and use another neural network, usually AlphaFold 2, to like predict the actual structure and to see if like the RMST between these two is low or not. So this is the main method of how we evaluate AI-generated structures right now. And then the other thing is like how diverse are you sampling your proteins as well? You don't want to just sample the same protein each time. That's not useful. You want to make sure that it's sampling the actual distribution of possible proteins and actually even go beyond them and sample novel proteins. So there's a way to cluster them by just taking two proteins, calculating like the RMST, normalizing it by some length, and then doing a clustering algorithm, like hierarchical clustering, to figure out how many clusters there are from your protein. And so like this, yep. Yes, yeah, sorry, I should have said that. Yeah, in silico means computational. It's like a fancy way of saying computational. Yeah, and so this is kind of this road. This was like back in 2021 into 2021. Things have gotten a lot better since, but just to show that kind of what we found as an initial step was that we're able to sample things that actually pass this 2 RMST threshold of like what you generate and what you predict. But what you see is that like as you get to longer proteins, the performance actually gets worse. So like higher here means worse. So I'm not going to belabor that point, but the most interesting thing to me was that we're able to actually generalize beyond the training set. So that by learning this like equivariant diffusion model, we're actually able to sample things that are very different than what it was trained on. So here, this is like the RMSD again. You kind of want to be as low as possible, but you also want to be low as possible on the y-axis because it's the similarity to the training set. So these are 2 proteins that the model generates. One is kind of short and one is kind of long. And then we look in the PDB, which is the data set of this most similar protein to this. And that's overlaid in gray, where you can see that these 2 proteins are extremely different and these 2 proteins are extremely different as well. So the model is able to kind of like generalize and learn beyond the training set, which is a very good indication for this model. Yeah, so summary with FrameDiff is that we kind of achieved 3 of the 4 goals, which is that we want to generate high quality structures that are diverse and novel, but we still have to yet design functional structures, which is going to be the next step of like how to actually do protein design with this. And I kind of left this out. I usually go over frame flow as well, but this is basically the flow matching version of FrameDiff, where we're using like an ODE to do diffusion, to do flow matching over SE3. And we actually get a lot better results with frame flow, and a lot of the current state-of-the-art methods are moving to flow matching. But just in the sake of time, I'm not going to discuss it, but it's something that I think hopefully you all should be very familiar with now. So I'll just take a quick break here to answer any questions about FrameDiff, if there's any. Yeah. Yeah, so at the initialization, you have to determine like how many tokens the model is going to have, and then you give it like this positional encoding, like positional encoding from like 1 to 200 or 1 to 400. So the model kind of looks at the positional encodings and decides, or like knows I'm going to generate a protein of this length. So it's a parameter you have to give the model. The model doesn't figure it out by itself of like what length it's generating. But there are works to kind of like have the model sample a length and then generate a protein with that length. Yeah. So my intuition for that is that I think flow matching is more general in a sense, and it works better for Riemannian manifolds. So in a Riemannian manifold, there's some assumptions there that around the space that you're actually diffusing over. And flow matching is a lot more amenable to those manifolds. So if you look at like SO3, like geodesics are very easy to define on Riemannian manifolds, but actually finding a noising kernel for Riemannian manifolds is not trivial. SO3 is one of those very few cases where you can actually analytically write down a kernel, like a noising kernel. But other manifolds, you're kind of sometimes out of luck for those. And so actually, the geodesic is like a much more stable thing to compute than like a noising kernel on a manifold. So that kind of goes into a little bit of the weeds there. But I think also like flow matching, because it's just like a lot more simpler as well, and it allows you to go beyond like Gaussian spaces quite easily. Yeah. Yeah, so the model learns to keep them close to each other. And this is why the positional encodings are crucial and why the attention mechanism is really important as well. It'll kind of learn that if I'm token 5, then I want to be next to token 6 and right after token 4. So it kind of self-arranges it as the diffusion process happens. Yeah, so RMSD is you kind of take the two proteins, you calculate the capture alignment after you center them, and then you just calculate the pairwise distances and then average that there. Yeah. Okay, great. So okay, now on to like more exciting stuff, which is like we've gone over unconditional generation, but now we want to do conditional generation. This is what people actually care about in protein design. So this is a paper that I collaborated with the Institute of Protein Design at the University of Washington, published in Nature, that we call RF diffusion for protein generation. So kind of the idea here is, like, I think we've all heard like pre-training, how important that is to language models. And so we kind of take the same idea to take a pre-trained protein structure prediction neural network and then fine-tune it with diffusion. And so the University of Washington folks, they had this pre-trained structure prediction network, and then we came along with like the diffusion idea, and it was kind of like a merge of two ideas that led to RF diffusion. And then the pre-training was actually very crucial because it kind of led us to train a lot shorter, and then also drastically improve this unconditional generation task, where the previous method kind of could only go up to like length like 300, but this model is able to go up to like length 800 or something, like, or even 1,000, and generate these like massive proteins that look really nice as well. And then this model is also able to be like even more novel. That like 0.5 is the cutoff point of like if you're below this, then you're quite dissimilar to anything in the training set, and the model is able to generalize even more beyond the training set as well. So this is something RF diffusion generated, and then this is the closest thing in the training set. You can see that the model kind of learns some of this pattern of this like sheet-looking thing, but then it's kind of able to extrapolate beyond that and generate new proteins from this. And then the most important thing about this model is that it was trained for conditional generation. So I'll go over a few of these. I'll go over symmetrical generation, where you add symmetrical noise and a model is going to generate a symmetrical protein. The most important is binder design, where you're going to tell the model, like, here's a virus, generate me a protein that binds to it. And you can also give it like a 2D like instruction that I want this specific looking protein. Or you can do like an in-painting task where you give the model a partial protein and ask it to generate the rest. And then the cool thing is that you can also combine multiple conditions together. So here we're combining both the symmetric generation and the motif, like in-painting generation, to come up with like doing both symmetry and motif scaffolding here. And then the other thing that's important here is also classifier guidance, where using like Bayes' rule, you can add these extra terms to kind of bias the generation away from what it was trained on, but towards like more desirable kind of proteins. And so, for instance, we might say, like, here's a molecule I want you to kind of encapsulate and bind around, and I'm going to create this potential field where if you're very close to the molecule, I'm going to try to repel you away from it. But if you're too far away, I'm going to try to guide you to get closer to it. And so then here's an example where these sort of issues might happen, that the model might generate a protein that might clash with a small molecule, but by applying this sort of potential, you can tell the model to kind of, like bind to the molecule like almost perfectly. And so here's like an animation of binder generation. What's happening here is that the model sees this protein, and we're telling the model to kind of bind like right above it. And so the model is able to follow those instructions and kind of generate a protein that's above there. And here's like another example where there's this existing portion here of the model, the protein that the model sees. It's going to try to fill in the rest, but also follow this symmetry constraint that it has to be symmetric, but also fill in the rest of this protein here. And then so for binary design, I kind of alluded to this before, but one important thing is that we have to give it these conditions of like where you want it to bind. So you can tell the model bind at site 1 or bind at site 2, and the model is going to follow these instructions to bind at the specific location. Otherwise, the model is going to bind at some random place that might not have an actual importance in the downstream application. But also, you could also condition on the fold topology, so like what kind of protein you want to generate. You can tell it both bind here, but also this is the kind of fold you want to be, or like the topology. You can also tell the model to kind of hallucinate and come up with like its own type of protein that will bind at that location. And then the coolest thing about proteins is that you can also actually test them out in the lab. So we took this to the actual wet lab, and there they actually measured how well the protein was actually binding to that. So not only is this, like protein that looks good, but it actually can be produced in real life. These are like 4 cases where, to show like the generalization, it can generalize across different targets and produce real binders. And then compared to like previous algorithms, like I was mentioning, this structure generation was the limiting factor. Those methods, almost had an experimental success rate of like 0.1% or something. But then with these diffusion models, we're able to go orders of magnitude higher than that, where for this one it's like 30% success rate. Yeah, all of these are like order of magnitude higher than what was previously possible. And then, yeah, this is like a actual structure that was calculated in the wet lab, where this is like a flu virus protein, and then we designed a binder that will bind to here, and they actually looked at the actual structure, and that the binder matches very closely with what came out of the AI algorithm. So this is like just further validation that the design and the real protein match there very closely. And then this is symmetric protein design, where there's a lot of symmetries in nature. This is like one way of how it stays efficient. That like a viral capsid, this is like kind of how the virus enters the body. We can like analyze the symmetry of this and realize that that this is made up of 60 different copies of this single protein, but just tiled up to follow this icosahedral symmetry. And then we can take the symmetry group and apply noise to the generation to kind of generate a protein that will follow this symmetry. So I think the animation is a lot better in the words. So I have this animation here of like how the noise is added symmetrically with like good starting positions of the noise, and the model is going to like figure out to generate a symmetric protein at the end. Okay, I think that's the end of that. Yeah, and then the cool thing again, of course, is that you can also measure these proteins. So these are like actual pictures that someone took of the proteins after they were synthesized. And you can see that the 3D volume of the proteins closely match what came out of Erives diffusion. And that's kind of remarkable that you can go from an AI algorithm to the actual protein and validate it. It's one of the nice things about this field. Yeah, so Erives diffusion follow-ups. One thing is that we're moving beyond proteins to also incorporate like small molecules and other types of non-protein things. And another thing is antibodies. This is like a very important class of antibodies that a lot of drug companies are very interested in. And so there's been work to specialize Erives diffusion to specifically make antibodies for therapeutic applications. So, so this is kind of a takeaway so far, that we've now accomplished all 4 of these with these past 2 works. I was going to go into a little bit into co-design, which is doing both like structure and sequence generation. But I think maybe I'll just take a few questions here in interest of time. Okay, so carry on forward. Yeah, so co-design. I should just have a few slides on this, which is, I was talking about this 2-step process where you would go from like a function to a structure and then to a sequence. And it's kind of, if it's happening sequentially, why not just do it together at once? And so in co-design, the task is to generate both the structure and the sequence at the same time. So there could be benefits of this, as in you might have like better control. You're also solving a chicken and egg problem, where do I do the sequence first or the structure first? So yeah, that's kind of the question that we try to answer. And so what I was working on last year was just come up with a formulation and the algorithm to do this. And so the algorithm we came up with was multiflow, where it's a flow matching algorithm over translations, rotations, but also the sequence. And I kind of described the translations and rotations previously, but a sequence thing is kind of a new thing where you actually have to describe a flow matching algorithm over discrete spaces. And that is kind of technically has all the same principles in continuous space, but also requires some new developments there to make it work. So I kind of left out the technical details, but kind of the general idea is that you want generation of discrete data, but you want it to be over continuous time. Usually in autoregressive language models, it'd be like left to right generation, but in proteins, there is no inherent left to right order, so you have to have an any order algorithm that can generate kind of things arbitrarily in any order here. So by doing this together, this is like a sequence and structure co-design algorithm that's generating both the sequence and the structure at the same time, starting from T0 to T1. That should, at the very end, have both of them completely filled in here. Okay, yeah, so technical summary of this work. I think proteins are interesting because it's highly multimodal, so there's a lot of interesting technical questions to work on. You're working with like Riemannian manifolds and Gaussian space, and also discrete spaces as well. And so there's a lot of work looking into kind of what's the best algorithms, what's the best models to like do protein modeling. And this also translates to other molecules like small molecules and RNA, DNA. And it's kind of like what's next. I kind of hinted at this, but AlphaFold 3 came out last year, where they also introduced a diffusion model to generate proteins, but also things that are not proteins, such as DNA, RNA, as well as small molecules. And one thing that I think one of the questions was that they also removed equivariance, so it kind of brings into question how important is equivariance after all, or the scaling laws can get away with that. So I think my prediction for the next year or two is that there's going to be this huge shift to move beyond proteins and capture all of molecules. And then the other thing that I think that's going to be very important is fine-tuning and post-training. We have ways to generate a lot of proteins, but then after we do some experiments on them, not everything works out or fails or succeeds. So we want to be able to iteratively improve from the experiments that we get and build a general model that will get better and better over time. This is kind of how ChatGPT kind of worked by aligning to human preferences. We want to be able to align our models to experimental data. And that could have large ramifications for new biology. It's like a reference page. Yeah, so I'll take any questions here, but this is like just contacts. My advisors again, Regina and Tommy. I think if you want to do some research in this space or looking to work on diffusion models for biology in general, reach out to me or our co-advisors, same co-advisors as Peter and Ezra. And if there's any like website, Twitter and stuff, you can reach out to as well. So here I'm also happy to take more questions if there are any. Okay, clap also.
All right, cool. So I'll make a final announcement, and then we're gonna end. So as you know, today was our last class. So I just wanted to say also in Ezra's names together, thanks a lot for taking this class. We had a lot of fun. I hope you learned a lot.